Good evening to everyone. So, welcome to day 12 session on T cell epitope prediction. Before going to T cell epitope prediction in this internship and certificate course on computer aided drug and vaccine design, can we recap the last session topics? So, what are the last session topics we have discussed? Yes, very good. We discussed something on vaccines and types of vaccines, and we have discussed about conventional vaccinology and what is reverse vaccinology. So, if we start with working on bacteria, viruses, and pathogens, any kind of parasites, whatever we take, and if we start getting a part of the material like a protein, part of biomolecules like proteins, or if the virus is getting killed, or if virus is getting inactive because of removal of pathogenic factors or virulence factors, so whatever it is, okay, so whatever we do, that we call it as conventional vaccinology. And in reverse vaccinology, where I have explained you that we start with the genome, we start with proteome, or we go with any other omics like interactome, etc. So if we start with the already existing data from the omics, and then if we just explore the candidates, the peptide candidates from that, then that's what we call it as reverse vaccinology. For reverse vaccinology, we have seen we have the well-known database is immune epitope database. In immune epitope databases, plenty of epitopes are deposited as data for various types of diseases. Okay, bacteria, viruses. And not only we have epitopes, that is peptide epitopes data in this database, we also have tools, multiple tools. And if you see this immune epitope database, we can able to see two types of vaccine design. What are the two types of vaccine design we have discussed in reverse vaccinology? Two types of vaccine design in reverse vaccinology. So this is my question. Actually, computational immunology is what we discussed as this reverse immunoinformatics, isn't it? Immunoinformatics, reverse vaccinology is one of the approach. Not just we have seen based on the types of cells. I just given the clue. Based on types of immune cells, we can classify epitopes as the epitopes can be very good. That's the answer. The epitopes can be classified as T cell epitope and B cell epitope. And also, on last session, even the participants interacted well when I asked you about what are APCs, antigen presenting cells. Examples you have provided are B cell, macrophage, dendritic cell. These cells have MHC molecules on the surfaces, that is, major histocompatibility complexes. Each and every person vary with respect to HLA phenotyping. Like if you go with the transplantation, any organ transplantation, tissue transplantation, obviously one has to remember that the HLA phenotyping should match, or else you can find the organ rejection. Even the transplantation is not possible at all. So HLA alleles vary not just with the person to person, but even in a family, if you see siblings, okay, twins or quadruplets, even for them, also you can see variation with respect to HLA phenotypes. So we can have the same kind of a sequence and structure for hemoglobin, myoglobin, or whatever insulin, whatever proteins we have. But the HLA proteins, I mean HLA genes of MHC molecules, that alone will vary with respect to person. Even for vaccine design, one has to concern on the HLA types. Based on HLA type only, we have to design the vaccine. So commonly, if you design a vaccine, it work for few people. May if I design a vaccine for South Indian, okay, it work perfectly for South Indian, but it won't. I can't guarantee for North Indian. And even if I prepare a vaccine, especially for Nadar community, Then whatever I synthesize the vaccine, that works perfectly for NADA, but I don't think for Chetia. I don't think for Redia, Reti, etc. So like this, even with respect to community, with respect to specific population of a country, everything is possible. Okay, so how to design precisely? Okay, that's what we call it as a precision vaccine design. In precision medicine design, either we can design the drug or we can design the vaccine. So even we can precisely can design the vaccine based on a particular community or based on particular population. Everything is possible. Tomorrow, I mean today, I'll be discussing on about this. Okay, how to design precisely for the population. Now, later, what else did we discuss? We discussed on the types of MHC molecules, isn't it? So the next question is this: types of MHC. From vaccine design onwards, you should have also study immunology. Okay, so earlier for the drug design and molecular mechanics, one should have a knowledge on pharmacognosy, pharmacology, bioinformatics, especially computational biology, cheminformatics. So these are the branches of study we have discussed. But if you come across vaccine design, the most important thing is immunology and immunoinformatics. These are the two things. Bioinformatics vary with respect to immunoinformatics. In immunoinformatics, we study only the immune proteins. In bioinformatics, overall biomolecules. Okay, so here we specifically study on immune proteins, especially that to MHC1 and okay. So you have mentioned like MHC1 and MHC2. Very good. The exact answer is 1, 2, and 3. The MHC 1, 2, and 3 is the answer, but the significant alleles are 1 and 2. So when we work on design, we generally focus on 1 and 2. And I also showed you what are the different HLA types for the MHC1 and MHC2. So all these things will be recapping today also, and then we can see some skills on how to work with this T cell epitope prediction. Now let me go ahead. So these are the contents. So today we'll be discussing on workflow, T cell epitope databases, types of MHC molecules, difference between MHC molecules, immunological database, like how to use this IEDB and how to use the other databases. What is T cell epitope prediction? I mean, what are the types of T cell epitope prediction methods available? So how about immunogenicity, conservation, toxicity analysis? We see extensively on COVID-19 vaccine adjuvants. You remember when I asked you yesterday, I mean, on the last session about how many vaccines are there in COVID-19? This is in 2020. I replied you that we have around 89 vaccines, and we have only popular three vaccines spread around in Indian markets. Okay, so lot of things are there. Now let me go ahead with this T cell epitope. If you go with the T cell epitope, okay, I told you here also we can classify it into two types. One is TH and another is the TC. And if you see this, okay, this CD4 T cell generally interacts with the MHC2 molecule of the APC. So other than this CD4 to MHC2 interaction, even the T cell receptor, there is one more receptor called T cell receptor. This also will interact with this MHC molecule. This MHC2 molecules, if you see, we on the surface where the antigen, this is what we call it as antigen. Whatever antigen is going to bind, okay, it is going to bind with two different units. One is alpha 1, the other is beta 1. So these are the two units which hold the antigen. You remember I told you in MSC1 pathway, I just explained you on MSC1 pathway that if any of the bacteria or viruses are engulfed by the antigen presenting cell, you know that antigen presenting cell like dendritic cell, macrophages, these can engulf the bacteria or viruses invading in our body. So whatever it engulf, yeah, it has the proteosomes. The proteosome proteins present in APC can degrade that virus membranes, and the proteins are released, and even the proteins are further degraded into small peptides, and these peptides are projected into these MHC molecules. It can be MHC1 or MHC2. You find a more or less same, but a slight difference is there. And if you see this, alpha 1 and beta 1, the antigen is present on the surface, and this antigen, whatever present in the APC, is going to bind with the CD4 of the T cell. Okay, and if you see, this is the CD4 receptor. So for the antigen presenting and processing, for antigen presenting and processing, okay, we have CD4 and we have T cell receptor and we also have MSC. So all these three receptors along with the CD80, we have a receptor called CD80. Okay, the CD80 or CD86. Okay, so these are, we call it as co-stimulatory molecules. These co-stimulatory molecules, if it interacts with the CD28, this is another receptor from from the T cell. So if CD28 of T cell interacts with the CD80 or 86 of co-stimulatory molecules of APC, you can see the T cell is going to get activated. The T cell is what is going to what is a process happen now? The T cell activation is done. Okay, after the antigen presentation and processing. So this is what processing. That is, antigen is interacting with the T cell. Whatever antigen present in the APC is interacting with the T cell. So if T cell is activated, what is going to happen? One T cell is going to proliferate into two T cells. And similarly, this T cells can also release small cytokines. Okay, can also release small cytokines. Whatever cytokines it release, this can also bind with the APC and activate the APC. If it activate APC, again you can find the same. The APC can also can proliferate into one cell into two APCs. So similarly, if you see the increase of immune cells in our body can directly go and combat the cancer. Some proteins produced from the cells can also combat the cancer. For example, tumor necrosis factor, the popular tumor necrosis factor. In this, we have a subfamily called alpha. Tumor necrosis factor alpha. Okay, the name itself shows that these are the proteins directly can combat the cancers. So if if you see the second thing, okay, that is a CD8 T cell. So this is going to interact with the MHC1 molecule. Where is it? MHC1 molecule. But if you see, the antigen is not binding with the two different subunits, but it is binding with the same unit, but we have two different parts in that unit, like alpha 1 and alpha 2. Okay, so this is, we call it as a macroglobulin protein. So whatever this alpha 1 and alpha 2, which is having this antigen on the surface, is going to interact in the same way. The only difference you can find is, instead of CD4, you can able to see the CD8 molecule. The remaining thing, CD28, CD80, 86 interaction, everything is same. One thing you should remember that I told you on last session. So the father of dendritic cells, Ralph Steinman, who worked in Rockford. Okay, he received Nobel Prize in 2014, and in 2017, two different scientists worked on the T cell activation and T cell suppression. So if the co-stimulatory molecules interact with CD28, I told you get T cell activation. In that case, this phenomenon, this mechanism we can use for treating the cancer. But instead of CD28, if you have another molecule called CTLA4, cytotoxic T lymphocyte antigen 4. So if this co-stimulatory molecules interact with the CTLA4, then obviously the T cell is not going to get activated, but it is going to get suppressed. This kind of a T cell suppression is generally used for treatment of autoimmune diseases. The mechanism of treatment of cancer is just quite opposite to that of treatment of autoimmune diseases. If T cells are suppressed, and obviously this interaction won't give a good kind of a cytokines, and these cytokines are not going to bind with the APC, and so just because there is a stipulated life cycle for the T cells and APCs present in our body, so obviously these cells will be dying just because of T cell suppression. Okay, so one can also check the life cycle of the T cells. Okay, produced and dendritic cells. Even the life cycle is just 9 days, I believe, 9 to 12 days. So obviously these cells will be dying, and obviously we can combat even the autoimmune diseases because of the CTLA4 molecule present on the T cells. It applies even for the CD8 T cell. Okay, and if you see this peptide, whatever I showed, it's very clearly mentioned. Just because we have two different macroglobulin, the size of the peptide, okay, whatever you can see, is maximum for CD4 T cell when compared to CD8 T cell. And these are the types of receptors. And I told you for this helper T cell, the CD4 T cell, also called as helper T cell, the, just because it's going to bind from here to here, so the minimum size of the peptide what we have to design is 13 to 17. But I prefer you to use 13 amino acid, because even in John Hopkins University, many of the scientists, medical scientists, use this kind of a 13 size for designing. Either fix one size and try to do this epitope design. And for the cytotoxic T cell, similarly here you can able to find around 8 to 12 amino acid. I suggest you to fix a size like 9. Okay, fix a size like a 9, and you can predict the thing. Okay, so this is how the we can able to predict the T cell epitope for helper T cell as well as cytotoxic T lymphocytes. To know about the cytotoxic and T lymphocytes difference, you can see further in it. Okay, so cytotoxic T lymphocytes directly combat the cancer cells, but the helper T cells can take help of other cells. Okay, and if you see this dendritic cell markers, I told you in my PhD research, I worked on 162 surface receptors of dendritic cell markers, but not all are significant with respect to T cell activation. I told you nearly three molecules. Only three molecules are majorly responsible for activation. So watch my earlier video about the T cell activation, and also just now also I explained you how to make it. This is a pathway for MHC1. MHC1 molecule receptor structures. I told you in MSC1 we have alpha 1 and alpha 2 of the same macroglobulin unit. But whereas in the MSC2, just because we have two different proteins, so only we need a lengthy kind of an epitope to be designed. But whereas here we just want a small peptide to be designed. And MSC genes and MSC functions, we have major the important genes like DP, DQ, and DR in MSC2. But whereas in MSC1 we have ABC. MSC3 is less significant because of complement proteins. It code for this is significant with respect to complement kind of immunity, complement molecule immunity. And we also have minor MSC class molecules like E, F, G, but in databases, rarely we can see this. Okay, we majorly see these three genes for MSC class 1 and these three genes for MSC class 2. So these are minor genes for MSC2, TM and TO. So in one chromosome 6, they have showed the locus of all these genes. Okay, in one chromosome itself, you can able to see all these genes. And if you see this HLA and how it links with the autoimmune diseases, HLA B27 allele is responsible for these three diseases, and B47 for 21 hydroxylase. So similarly, you can see the MHC2 genes and its relation with respect to diseases, that is with the increased risk. If the community people have this gene, obviously they are prone to get these kind of diseases. Okay, so this is something which is related to HLA and autoimmune. Similarly, you can even see for the cancers. Now, Raghava web server, I am showing you another web server other than IEDB. So I told you on last session itself, this Raghava is from IIITD. So he is from this institute, and of course, in this his organization, he has developed multiple tools and databases for the T cell helper prediction, epitope prediction, cytotoxic T lymphocyte prediction. We also discovered few more tools for immunoinformatics. So for T helper cells, we have this much number of tools present, and these are the tools. Other than IEDP, you can also try with it. We can't try with all this, but we can take some two or three and we can compare with the IEDP database. Whatever we are getting the epitopes, whether the same epitopes are even generated from this or not. So we get checking the coincidence, okay, in this Raghava Web Server database. In ProPred, this is how the homepage of ProPred looks like. And this is the URL. Okay, in Google, if you simply type ProPred, it directly takes you to this. In ProPred also, we have two types of servers, ProPred 1 and ProPred. ProPred 1 stands for MSC Class 1 Binding Peptide Server. And this MSC Class 2 for ProPred. We have two different URLs for this, but both are developed by the Raghava scientists. And if you see this here, the input what you have to feed is a sequence here. Okay, you have to paste the amino acid sequence of the viral protein. For example, if I take Nipah virus, I have already circulated a paper to you, a journal paper. So whatever paper I circulated and I explained in the last session, I told you only one protein. No scientist have worked. Myself, I have taken this pathogenic protein W and started working on both T cell epitope prediction and B cell. So T cell alone I have published. And if you see this, we have to take this protein sequence from UniProt. Okay, initially, if to work with this kind of a prediction, take that Nipah virus W protein from UniProt and paste the sequence here. Whatever that W protein sequence you pasted, now you have to select the appropriate alleles. Which are what alleles are most important? I mean, if you're targeting the Indian population, so what are the major alleles present in Indian population? That you can select. You know, the definitely the Indian alleles vary with respect to American alleles, and of course it varied with respect to Australian alleles. So each and every country, the persons have a unique specific kind of alleles. So whether you want to design for Indian population or Tamil Nadu population, Andhra population, we can check that information even from IEDB. That information is available. Allele information checking is available in IEDB. And after checking that population allele information, we can try the same here. Okay. So this is something like initially you have to try with IEDB, then you can cross check with this ProPred servers. Okay. So these are the exclusive servers for MSC1 separately and MSC2 separately. For MSC1, I already told you, better try with 9 amino acid peptide. Okay, and for MSC2, better try with 13 amino acid peptide. You fix one length and then you start predicting based on that. And net MSC PAN 4.1. This is another kind of a thing from Denmark. Okay, so they have developed these kind of a server. Here also you have to paste the FASTA format of the sequence like Nipah virus W protein sequence, and then you can start checking for this MSC epitope prediction. Net MSC PAN 4.1. So if you see, if you drag down, you can find this. Here only you have to paste the amino acid sequence. Then you have to click submit. Okay, this is how an IEDP website looks like. I told you to look through this database if you have time. Even yesterday I've given time, isn't it? I think many of them have tried this. So there are peptide epitopes, non-peptide epitopes. So these are the numbers already deposited. Okay, the epitopes whatever deposited are around 8 to 20 amino acid peptides. Okay, in different lengths, you can see the peptide epitopes are deposited for all the types of diseases. Okay, infectious disease, allergic, autoimmune, asthma. Okay, what the epitopes vary with respect to the disease. Some epitopes are specific to the disease. So, if you want to check epitope for a specific disease, we can check with this. Similarly, we can try with T cell separately, B cell separately. You know how these epitopes are deposited? Obviously, through assays. In vitro experiments are done, and through in vitro experiments, they whatever results they get, they got this kind of a thing. And also, we use a computation algorithm to generate these kind of peptides. So they cross check with it, and they have deposited in this database. Okay, and not only that, we also have different types of host. Not only it is useful for the medical patients. Okay, it is also useful for the veterinarians. The veterinarians can use can also predict vaccine for the animals. Can be the dog, camel, that is cow, buffalo. Okay, also we can check with the wild animals also. All the kind of information is available in IEDB. T cell epitope we have exclusive, T cell epitope exclusive, and epitope analysis tools. Where today we'll be discussing on how to use T cell epitope and epitope analysis tools. MHC class 1 tools. Just if you go to this MHC 1 from IEDB, you have to input the amino acid sequence of the protein. So here I have input the amino acid sequence of the protein. After that, I have to fix the peptide length. The peptide length I fixed is 9 amino acid. Okay, because if you change it to some other length, then obviously you can see the peptides vary. Okay, so fix one length and you can use that as a control and you try to complete the experiments. MHC alleles you can select from the database present in IADB. Okay, and then we have to click simply submit. And similarly, this is for MHC2. Okay, so the similar kind of window will be there, but fixed length, either fixed length like 15 or 13, whatever you want to try with it. So for example, I tried with 15, I got some kind of results based on that. I told you, like even we can design epitopic based peptides based on specific population. So to go to the specific population in the IEDP database itself, we have like tools. In the analysis tools, one has to go through the this population. Okay, so there is a database called population. Here you can see all the population are available. East Asia, Northeast Asia, Northwest Asia, South Asia, all these kind of Asian regions are available. And other than that, you can also see Australia, America. So if you want to design a vaccine for a specific population, you can also use this to check. Okay. And obviously we have, this is the most important thing. We have an hyperlink called allele frequency net database. So in that, we have to go to this allele frequency net database. If you click this hyperlink, it takes you even to the community. That is, if you want to design a peptide with respect to region, you can use this. But if I want to use with respect to the community, like NADAR community, REDIAR community, then obviously we can use this allele frequency net database. Okay. And after that, you have to feed the epitope followed by MS restricted alleles. Okay, so to analyze the results, and this is the application I mentioned for practice. Okay, so also published a book chapter related to this. So I told you the workflow on the last session itself. So I take an epivirus protein W sequence and then I start predicting the epitope from my EDB. Then I go with docking and then simulation. So simple process. So Nipah virus genome. So because I just going to explain a demo with the Nipah virus. So some information on Nipah virus. So in Nipah virus, we have again the two types of proteins like structural proteins and non-structural proteins. You remember in SARS-CoV-2, I have mentioned you, we have 4 structural proteins. A non-structural, we have 16 proteins. But whereas here, non-structural 3 proteins. C, V, and W. Whatever I worked is just non-structural protein W. But all these proteins are already worked by the scientists and published in repeated journals. Okay, so if you see structural, 1, 2, 3, 4, 5, 6 proteins are there. So Nipah virus genome. Nipah virus genome obviously varies with respect to SARS-CoV-2. SARS-CoV-2 on the surface itself have spike, so we call the spike is one of the major target because of present on the surface. But Nipah virus is something different. Okay, we have both structural, non-structural, and both the structural, non-structural are significant in target. Okay, so this is something about Nipah virus genome. Now about T cell epitope prediction. So how this epitope prediction is done in computational methods. So what is the logic used behind? If you see, first it classify this T cell epitope methods as direct methods and indirect methods. What is direct, what is indirect? Now we can see one by one. If you see this direct prediction procedures on T cell epitope prediction, these features uses ampipathicity, that is hydrophobicity, hydrophilicity, and then motif. Some proteins have both the kind of hydrophobic and hydrophilic features. Then obviously we can find that kind of a future. And similarly motifs. So not the entire protein is responsible for a immune function. A part of a protein, that's what we call it as a motif. That motif, whatever it binds MSCs, that is most important. Okay, the T cell, I mean, that viral protein motif that binds with the MSCs. But they are quite inaccurate and high false positive. Okay, and there are something called indirect methods, which uses machine learning techniques like artificial neural network, support vector machine, and the other theories. Okay, one more thing you have to remember, other than direct and indirect prediction, we also have prediction. We can predict the cleavage and transporter associated with the antigen processing. That is what we call it as STAP. I told you in MSC1, MSC2, we have 9 molecule and 13, I mean, 9 amino acid length peptide, 13 amino acid peptide binds on the surface. How this is binding, how this is transporting. So, for the transporting, this is a different function. So, for this different function, we can also predict in the IEDB itself. We have a TAP tool available in IEDB. So it tells you about the presentation and processing of T cells, the TAP tool. And now let me go to the next concept, T cell immunogenicity. So in T cell immunogenicity, we can able to predict one is antigen processing, number two is peptide binding to the MSC molecules, number three is recognition by a cognate T cell receptor. This is what I call it as immunogenicity. Okay. And if you want to work with the immunogenicity, we can go with tools edb.org/immunogenicity. This is the URL. And conservancy analysis. The conservancy is a fraction of the protein sections that the epitope contains an identity is a similarity. So if you see this epitopes, the epitopes can also be classified into linear analysis. And so this is one of the conservancy epitope. And number two is the non-continuous sequence, linear and non-continuous. So this you can able to check with this tools.immuneepitope.org. And toxicity, we can check through these parameters like hydrophobicity, hydropathicness, that is hydrophilicity, molecular weight, net hydrogen, side bulk, and steric index. You remember in the beginning of the session also I told you we use one tool called Protox3. The Protox3 generally used for determining toxicity for small molecules. They also that kind of a server also uses these kind of a properties for prediction for small molecules. Here, if you want to predict toxicity for peptides, we are using this Raghava web server, toxin pred. In this toxin pred, the calculation, okay, the toxicity calculation is done through these physicochemical properties of the peptide. Now, how to work on the project, how to work on a research. So initially, you have to take the FASTA format of the protein sequence from the UniProt KB. UniProt is the overall URL. So in UniProt, we have UniProt KB. For fetching just the FASTA format, you can also use UniPark. It means that Universal Protein Archive. Okay, even there you can get the FASTA format. Whatever FASTA format you take, you have to feed in MSC1 server and MSC2 prediction server. Similarly, you can use it same input for TAP, transport associated proteins, for knowing about antigen processing. Now, whatever we take this amino acid sequence of viral protein, this can also be used in other databases. Other than IEDB, we also have ProPred, NetCTLP, PAN, NetMSC PAN, Predicted T cell. So these are different servers. Even through that, we can able to predict. Okay. So this is something about this. Actually, this is actually the scores are like this. Okay. Yeah. So if you see this kind of thing, okay, actually, I've done a small, uh, there is some mistake is there in my research, but still it got accepted. I don't find any previous or pinpointed that. If I want to work with MSC1, I told you if you have to fix with 9 amino acid and MSC2, 13 amino acid. You know what I did? I fixed commonly as 9 amino acid for both MSC1 and MSC2 and I published. But if you want to work, you will work correctly. Okay, 9 and 13. That is a preferred thing. Or you can use a 15 amino acid for MSC2. So we can predict separately. Okay. So according to the research, whatever I worked, uh, I got this is the best peptide for MHC1, and this is the best peptide I got for MHC2 molecule. So I predicted only one base peptide. You know how this peptide is predicted now from the series of peptides. So for these peptides, whatever I've showed, okay, out of thousands of peptides for the Nipah virus W protein, I have screened the base peptide based on the consistency in this increase in score, and also based on the immunogenicity, conservancy, and toxicity. If you see this immunogenicity of this peptide, okay, SPV for MHC1, whatever I screened, okay, so this is actually showing a good score, a top score when compared to the others. Okay, other than immunogenicity, even conservancy is good, and toxicity didn't show any toxic. So just because it is showing a significant kind of a result, and of course, this is okay. They are not bad. Something like that. We got as scores from two different servers. Where I just screen this as a base epitope. So like this, we can use our methodology, an algorithm to fix a base peptide epitope from thousands of epitopes generated from the server. And similarly, for the MSA2, I have screened this peptide, and this peptide, whatever I am showing you here, okay, showed a good kind of a conservancy, non-toxic, and even this value is positive maximum. So like this, I have screened these peptides. So whatever peptides I have screened for MHC1 and MHC2, this is an MHC1 peptide, and this is the MHC2 peptide. Okay, so that is what do you mean by MHC2? These are HLA, this can be A, B, or C. MHC, no, sorry, this is a 2, isn't it? 2, not DP, DQ, TR. It can be any of the genes. I tried with DR. I tried with DR because I focused only on Indian population. If you want to focus on a specific community or population, we can select these alleles. So this sequence, this amino acid sequence, I have taken from the NCBI, and I have modeled this protein using Swiss modeler. Whatever I modeled protein from Swiss modeler, I try to talk with my peptide using these servers. And similarly, here peptide protein docking server is used to dock the predicted peptide versus HLA P molecule. So molecular simulation, I told you after docking, we have done the simulation, and I told you how to analyze the simulation. So to analyze the simulation, we have to check the consistent pattern. Okay, so if you see this, it comes around 0.75. So up to these residues, this is stable actually. Okay, up to these residues, this is stable. You can tell the time and thing. If this is for root mean square deviation, for root mean square fluctuation, I told you the fluctuation should not cross 1 angstrom. Fortunately, we didn't find 1 angstrom and above 1 angstrom. So this is actually stable, okay, with respect to C alpha fluctuations. Whatever fluctuation I'm telling is related to carbon alpha fluctuations. The carbon alpha is present in each and every amino acid, isn't it? So these amino acids residues are mentioned in this x-axis. And you can tell if there is some kind of a fluctuation, this alone you can report. But whatever fluctuation you have, this is insignificant. This is not a significant one because it's less than 1. Okay, 0.57 something is less than 1. And then we also worked with the area. The area we have to check consistent area. Okay, what is a consistent area for a period of time? So for this period of time, you can see the area, and similarly radius of gyration. You can check the consistent. This actually I told you this can provide you a data on moment of inertia. Okay, so the molecular simulation tried with both the molecules and reported in this journal paper. And I also mentioned you about the vaccine adjuvants. In my lab, in my research lab, for the past 15 years, we are working on vaccine adjuvants, development of vaccine adjuvants. Myself, I worked with IL-2, IL-4, GM-CSF, AH03, dendritic cell as an adjuvants. And presently, we are my startup is synthesizing around 26 different secondary metabolites from 6 Indian medicinal plants. So most of the secondary metabolites can also be used as an adjuvant in future to replace the aluminum salt. Aluminum salt is nothing but an inorganic chemical. This inorganic chemical going to accumulate in our body and it gives some side effects. To reduce the side effects, we are using this biodegradable kind of materials, biodegradable kind of biomolecules. Okay. And this is the database we have developed, whatever the database we have developed in Stephen Venkateshwara College of Engineering. This can be used to predict a plant-based adjuvant for a specific type of diseases. If you select a disease, it can tell you this is the best plant adjuvant. If you select another disease, it give you a different plant-based adjuvant. So this is done actually by depositing the docking data of plant molecules with respect to surface receptors of dendritic cells. And this is, of course, a part of my PhD research. I majorly worked with two different plant molecules, Pyronon B from Pyrevia diffusa and Vithnot A from Withania somnifera. These plant molecules triggered differentiation of immature dendritic cells from monocytes. And not only that, these molecules also can able to mature the dendritic cells. If this happens in a quick process, then obviously this T cell activation and producing of other cytokines cells, this will be more rapid. And you know that if you want to go with the treatment of cancer, this cancer is nothing but abnormal cell proliferation. But if our immune cells can proliferate faster, these immune cells can combat the abnormal cells. Now, related to adjuvants in COVID-19 vaccines. In COVID-19 vaccines, okay, polyethylene glycol is used in mRNA spike protein, and adjuvant like adenovirus is used in AZ one, AZD1222 virus spike protein vaccine. Covishield, even myself, I have taken Covishield. Many of them have taken this. Here, adenovirus, actually, whatever adenovirus vectors they use, that itself act like an adjuvant. And so in the early cases, it's just 70% affecting. And if you see this COVAX, that is something showing a good kind of an efficacy. It depends on the patients, whether you're treating the Indian patients or treating South Indian patients, North Indian patients, everything. Okay, the efficacy varies even with respect to alleles. Uh, and then if you see Sputnik, a Russian-based vaccine, assure a good kind of efficacy when compared to Covishield and Covaxin. So, but also see the adjuvants. In COVAX, a matrix M adjuvant is used. That is saponins. In COVAX, plant-based adjuvant is used. So you have to take this important thing. Like this, you can see in other COVID-19 vaccines also they have utilized the plant-based adjuvants. Okay, and similarly Sinovac, and you can see here functional diversity in the adjuvants. And in the second wave of COVID-19 disease, you might have seen so many kind of viruses, strains of SARS-CoV-2 came to existence. So for each and every strain, we can see out of 1,260 or 90 amino acid, I forgot that. This is the size of the spike glycoprotein. If this is the size of the spike glycoprotein, okay, if one amino acid, that is leucine 452, is mutated to arginine, then it gives a different strain. So like this, we have hundreds of strains of SARS-CoV-2 because of the mutations. So the theory part is completed. Now let me take you to the explaining on skills. How to work with a T-cell epitope-based vaccine design. So for this, initially we have to go with IEDB, and of course we have to go with UniProt W. If you have a protein sequence just like that, we can copy and paste the sequence. Whether I have any amino acid sequence, I just checking. No, I don't have. So then I have to find from the UniProt. So UniProt, okay, I use UniProt KV, or just go to UniProt. Excuse me. So UniProt, Nipah virus. So I just type like that, or you can type like Nipah virus W, however you want. So fetching is difficult. I'm getting something else. Can I type W now? So now I check at the top. Nipah virus MN is to LF, glycoprotein, and nucleocapsid protein. So we have W, something like that. So better go through that UniProt ID. I have already mentioned the UniProt ID here. If you know the UniProt ID, no problem. P0C1C7. Okay, P0C1C7. Yes, Nipah virus W. Go to the sequence. And this is the sequence. So download this sequence. Whatever download, we just have to copy. And whatever we copy, we have to go to the MSC1 or MSC2. First, I try with the MSC1. So I go to the MSC1 and then I'm going to paste this. So if you want to, default the SARS spike glycoprotein is there, but this is a very large size. Instead of that, I'm going to try with my sequence. So paste the Nipah virus W. After the default, it is in 9. But before that, if I want to focus on specific community population, for example, Tamil Nadu Nadar population, first go to the population coverage in the epitope analysis tool. So if you click the population coverage in epitope analysis tool, it yeah, it leads to a window where you can able to see the country-wise population. Okay, country-wise, whether if you want to predict country-wise specific or if you want to go with the community-wise specific, you can decide. Okay, so query by country. Okay, but I don't want to query with country. I want to go with allele frequency in a database. So inside that, I'm going to the allele frequency. So here you can able to see the country, but I don't want to choose. I want to go specifically. So if I just because I clicked allele frequency net database, it has taken inside. So after going to the allele frequency database, you can see this population studies. The next thing, what you can try, the allele frequency database, you can try with the HLA, or you can try with the populations. I click populations, you can see browse populations by geographical region. So click this. So under population, I'm going inside the geographical regions. So inside the geographical regions, first you can select the country, then the community. So if you see India, okay, India comes under South Asia, I believe. Okay, so I go to the South Asia. If I go to the South Asia, yes, India comes under South Asia. India, Andhra Pradesh, Bikaner, Delhi. So I want to first focus on the Tamil Nadu Nada, whether we have, or Tamil Nadu Chennai. This is best, isn't it? But Tamil Nadu Chennai, most of the Tamil Nadu Chennai, Chennaites have this TRP1 molecule. TRP1, it means that MHC2 allele. Okay. So if you want to go specifically this Tamil Nadu Chennai, I'm going inside the Tamil Nadu Chennai, and here you can see this allele frequencies. Okay, click the allele frequencies. You have to go inside to fetch the correct information. So if you see this DRP1, we have plenty of suballeles. DRP1 is the major allele. Inside you have 0, 0, 1, 0, 3, 0, 4. But whatever I tried, this is actually class 1. So if only if you try with the class 2, if only if you try with the class 2, so I'm just clicking class 2 now. I'm going to paste that Nipah virus. Here it is 15 amino acid, but if I want to fix it as 13, you can fix 13. Just because, yeah, if you want to get the 13 alone, I made it as 13. Now we have to fix an alleles. So what allele you want to provide? So according to this allele frequency, if you see which is the major according to the percentage of individuals, the major is DRP1 13, okay, and DRP1 3 or DRP1 4. DRP1 4, 4 and 7 are major. Can I give 4 and 7 then? So how to give 4 and 7? Go to the this thing. Okay. So HLA. This is MHC2. No, this is MHC1. Where is my MHC2? This is my MHC2. HLA DR. What we have decided? 4. 4 is selected. And if I want to select 7, HLA TRP1, I'll be getting. If you want to select 4 and 7, I just selected 4 and 7. Okay, whatever you want to select based on thing, you can make it. And then we can have to start predicting. To start predicting, simply you have to click prediction. MSC add another prediction. No, not needed. So simply click this run button. We have a run button here. I just clicked it. So now we are determining the MSC binding illusion. But see, this Chennai, it is have this MHC2 allele alone. But if I want to work with the MHC1 allele, what specifically I can work? Come back. Instead of Chennai, we can go with the C here. The Nadar is having this HLA, uh, uh, that is MHC1 allele, ABC. I go to the Nadar now. Inside the Nadar, go to the allele frequency. Inside the allele frequency, we can see the major allele frequency is 030101. Can I fix this? For fixing this, we already have this. Okay, what is that? We got 030101. So 03. First, I have to type like hyphen A asterisk 03. Okay, if I click 03, I have something like 1, 2, 3, 4, something like this. If I want to select 1, I can select 1. And if you want to select one more allele, you can select that. Whatever we have selected, simply we have already pasted the amino acid sequence, fixed 9 amino acid for MHC1. Simply click this run button. So in one side, we are predicting the MHC2. In another side, we are predicting the MHC1 allele. So what happened with MHC2? Yeah, we got the result. For MHC2, whatever we have predicted in IEDB, you can able to see this result. This result, if you want to download this in the form of TSV or CSV. Both can be opened in Excel. Okay, anything you can download. Can I try with comma separated value file? Because TSV already tried with the Cytoscape string DB database. So this time I'm saving it as comma separated value file. I'm going to keep it in bioinformatics demo CADVD December. So this is for actually MSC2. Okay, whatever we predicted for a Chennai it is. Okay, so I just downloaded this. So if you want to open this, you simply double click and open in the Excel. So better I just have a word wrap and increase the font. See here, these are the peptides actually. So in this we have the peptides, and this is the start and end length. So all are 13 amino acid length. Just because we fixed 13, we got this length. And these peptides are ranked based on this score. If the score is more, then that is a good peptide. But which is best, for the because if you see this kind of a result, we have how many peptides? This has generated 177 peptides. For the Chennai population people, we got 177 peptides, but we have to screen only one which is best. The only one which is best we can predict from this server based on the score. If the maximum is the most is the best one, but it is not sorted accordingly. If you so you can select here data filter. Now you have to see which is having the highest score, isn't it? So I deselect everything and whatever at the bottom I selecting this. So this is the best peptide. So this is the best peptide you can show 9 amino acid peptide. Okay. So this is for actually we are predicted for MSC2. Similarly, we can able to predict for MSC1. MSC1 is also predicted. We got this kind of a table in for 9 amino acid peptide. So now I'm going to go to download. So all rows in CSV file. The CSV file, whatever predicted for MSC1. Similarly, you can use the filter and you can find which is the showing the best score. So this is how we can screen MSC1 and MSC2. Okay, so if you want me to show, I'm just double clicking and showing it. So I just going with the wrap option. Okay, and then I'm increasing the font and I'm showing you the score like 0.8 something in random scores are there, but I keep a filter and I'm going to take the top score, the maximum score. The maximum score we got is 0.889. Okay, this is the thing. So this is our best peptide, whatever we predicted for MHC1. This is for Nadar community of Chennai population. So like this, So whatever peptide, so now you can understand. Then think about the SARS-CoV-2. Oh, each and every person is taking same Covishield, same Covaxin, and that too, we are taking the same vaccine for all the strains of SARS-CoV-2. Then how come we can treat effectively? So the efficacy is very less. So only you can see percentage of fatality is more, but at least Indians survived when compared to the Americans. So just because of specificity and other factors, okay, the alleles are most important, whether the alleles can manage or not. Okay, can manage this respiratory infection or not. That is just based on the human alleles. Okay, so allele finder is also available in this IED. Okay, so this is what how to use the MSC1 prediction and MSC2 prediction. Even in MSC2, we can predict immunogenicity separately, binding elution separately. Here also, I can predict similarly like that. And next to that, if you want to see what else is left out, yeah, ProPred. I told you ProPred, isn't it? Go to the ProPred and feed the kind of an inputs like ProPred. You can see if you have the same Nipah virus W protein you have to feed, and you can select whatever alleles we want. Okay, and then we can use something like summit sequence. We can try with it. We can also get a similar kind. So whatever peptides we get from here can't be matched exactly. Instead of getting exact kind of an output, we can. I suggest you to go with the best 10 peptides from MSC1. Or if you're trying with MSC2, try with MSC2 best 10 peptides and check whether these peptides are present in this peptide or not. If peptides are present, then you can able to tell that this is the best peptide matching with the two servers. So this is how we can able to make with it. Okay. So like this, we have plenty of tools available. Not only that, if you want to go specifically with respect to specific disease, even that kind of an allele information also we can able to find. Okay. For example, asthma is already given. I'm going to give something like asthma. We have types of asthma. Chronic asthma is advanced stage of asthma, isn't it? A lethal, is a dangerous respiratory infection. So related to asthma, you have to again type asthma. Okay, in the disease name, search related to that because we are getting so much information. Next to that, you can see about allergic asthma, chronic asthma. Again, you have to go here. So if you go inside, you will be getting even that information of the specific thing. And also epitopes can also be, it is directing to the disease ontology website, but also we can find the peptide information for the specific diseases. Okay, peptide information can also be retrieved from this. So asthma, chronic asthma, extrinsic asthma, allergic asthma. So this is a branch. So Moroccan allergy. So you can get this information related to that. Even peptides we can find from here. The epitopes are predicted for that. Okay, so whatever we predicted or from this assays, only one epitope predicted. Huh? That's what we got now. Okay, so later like this, you can able to find the information. So you can check for any kind of a specific diseases inside this IEDB. So IEDB, again, I'm telling you, it's an immune epitope database used for checking the T cell epitopes and finding the B cell and T cell epitopes, and to check with. So tomorrow we'll be discussing on how to work with B cell epitope prediction and how to work with work on immunogenicity and how to calculate toxicity. Okay, all these things we'll be discussing it on tomorrow. Today I have completed how to do T cell epitope prediction with IEDB database. So any doubts you can ask. Do you have any doubts? And even linear peptide, that is MHC1, MHC2 is a discontinuous. Okay, the linear and discontinuous we generally see in the B cell epitope, not the T cell. So we'll be discussing when we discuss on the B cell epitope prediction. Okay. So any doubts? Is designing vaccines also follows Lipinski rule 5? No, definitely not. Only for small molecules it applies. Any other doubts? If no doubts, thank you very much for the patience and interaction. So tomorrow we complete the B cell epitope prediction. Have a great day to you all.